Hello guys, welcome to the amazing world of biological molecules. In this video we'll uncover how these invisible molecules work together to keep you alive and healthy. Get ready to discover the hidden chemistry of life. We are going to discuss the SLO regarding structure of disaccharides and polysacharides. Let's dive in. Starting with the structure and illustration of disaccharides that are maltose, lactose and sucrose. As we have discussed in previous videos that disaccharides are formed by condensation and hydrolysis reaction. In organic chemistry, a condensation reaction is a type of chemical reaction in which two molecules are combined to form a single molecule, usually with the loss of a small molecule such as water. If water is lost, the reaction is also known as a dehydration synthesis. In a hydrolysis reaction, a larger molecule forms two, or more, smaller molecules and water is consumed as a reactant. Hydrolysis involves adding water to one large molecule to break it into multiple smaller molecules. First, the formation of maltose. Maltose, a disaccharide, is formed when two glucose molecules bond together via a dehydration synthesis reaction, specifically a glycosidic bond between the first carbon, C1, of one glucose and the fourth carbon, C4, of the other. This process results in the removal of a water molecule and the creation of the maltose molecule. Second one is lactose. Lactose formation involves the joining of glucose and galactose molecules through a glycosidic bond. Specifically, it's a beta-1,4 glycosidic linkage, where the first carbon of galactose is linked to the fourth carbon of glucose, with a bridging oxygen atom. Last one, the sucrose. Sucrose, commonly known as table sugar, is formed by joining glucose and fructose monomers through a glycosidic bond. This bond is formed between the first carbon of glucose, alpha-glucose, and the second carbon of fructose, beta-fructose, creating a unique ring structure. Next up in our video is formation of polysacharides. Polysacharides are major classes of biomolecules. They are long chains of carbohydrate molecules, composed of several smaller monosaccharides. These complex biomacromolecules functions as an important source of energy in animal cell and form a structural component of a plant cell. Some major type are amylose, amylopectin, cellulose, starch and glycogen. Let's discuss starch. Starch molecules exist in two forms, a simple linear polymer called amylose and a more complex branched form called amylopectin. Amylose is a linear polysaccharide chain that is made up of glucose monomers joined by 1,4 glycosidic linkage. Amylose makes up to 30% of the total starch molecule. It has a helical structure and is more resistant to digestion. Amylopectin is a highly branched polymer made up of glucose subunits. It is made up of linear chains of glucose units that are linked by 1,4 glycosidic linkage along with a number of side chains that branch the structure by 1,6 glycosidic linkages. Cellulose is another polysaccharide, and it is a major component of plant cell walls. It is formed by beta-glucose monomers linked by 1 to 4 glycosidic bonds. Cellulose is completely permeable, meaning that it allows water and other substances to pass through it into and out of the cell freely. Next is glycogen. This polymer of glucose residues linked by 1,4 and 1,6 glycosidic bonds. It is found in the form of granules in the cytoplasm. Liver glycogen acts as glucose reserve that hepatocyte release when there is a need to maintain a normal blood sugar levels. In the end, let's relate the structures of starch, amylose and amylopectin, glycogen and cellulose molecules to their functions in living organisms. Linkage type comparison. Starch and glycogen use alpha-1,4 and alpha-1,6 glycosidic bonds, while cellulose uses beta-1,4 glycosidic bonds. Difference in branching. Starch, amylopectin, and glycogen are branched, allowing for efficient storage and rapid release of glucose. Cellulose is unbranched and forms long, straight chains. Difference in function. Animals can digest starch and glycogen due to the enzymes that can break down alpha-1,4 glycosidic bonds, but they cannot digest cellulose because they lack the necessary enzymes to break down beta-1,4 glycosidic bonds. That's all, my dear students. I hope this lecture helps you understand the concepts. Give your feedback in comment section. Don't forget to like and subscribe.